Hi everyone, today we are going to talk about the enzymes. As we know, enzymes in the catalysis of biological systems are remarkable molecule devices that determine the patterns of chemical transformations. In this chapter, we're going to introduce enzyme briefly and its thermodynamics. So as features shown here, you will see a jellyfish, and actually, the activity of an enzyme is responsible for the growth of the luminescent, like it showed up luminescent jellyfish. So the enzyme we called equorin catalyzes the oxidation of a compound by oxygen in the presence of calcium to release CO2 and light. People find that nearly all known enzymes are proteins. There are only a few RNA among them. The most striking characteristics of enzymes are their catalytic power and specificity. So firstly, let us see their catalytic power. We show a table here, and you will see the enzymes could accelerate reactions by factors of as much as a million or more. So let us consider carbonic anhydrase, for example, because it is one of the fastest enzymes known. You can see that the transfer of CO2 from tissues to the blood and then to the air, in the alveoli of the lungs, would be less complete in the absence of this enzyme. So each enzyme molecule can hydrate 10 to the power of 6 molecules of CO2 per second. This catalyzed reaction is 10 to the power of 7 times as fast as the uncatalyzed one. Then let's talk about enzyme high specificity, including both in the reactions that the catalyze and substrates. So from the frame shows here, the biochemical function of these enzymes is to catalyze proteolysis, the hydrolysis of a peptide bond. And an enzyme usually catalyzes a single chemical reaction or a set of closely related reactions. On the other hand, proteolytic enzymes differ markedly in their degree of substrate specificity. For example, the digestive enzyme trypsin is quite specific and catalyzes the splitting of peptide bonds only on the carboxy side of lysine and arginine residues. However, thrombin is an enzyme that participates in blood clotting. That is just to catalyze the hydrolysis of arginine-glycine bond, in particular peptide sequence only. Of course, the specificity of an enzyme is due to the process interaction of the substrate with the enzyme. Besides enzyme, the catalytic activity of many enzymes also depends on the presence of small molecules termed cofactors. An enzyme without its cofactor is referred to as an opposite enzyme. So the complete catalytical active enzyme is called a holoenzyme. Cofactors can be subdivided into two groups. So 1 metals and 2 small organic molecules called coenzymes. So the detailed information for cofactors you can find in the table. Next, as you know, a key activity in all living systems is the conversion of one form of energy into another, and the enzyme plays the important role in energy transducting processes. For example, in cellular respiration, which takes place in mitochondria, the free energy contained in small molecules derived from food is converted first into the free energy of an iron gradient and then into a different currency, the free energy of adenosine triphosphate. But The molecule mechanisms of these energy transducting enzymes are still being revealed. Although enzymes speed up the rate of chemical reactions, the properties of the reaction, whether it can take place at all and the degree to which the enzyme accelerates the reaction depends on energy differences between reactants and products. And the Gibbs free energy, short for G, is a thermodynamic property that is a measure of useful energy, or the energy that is capable of doing work. It is a useful thermodynamic function for understanding enzymes. To understand how enzymes operate, you can say from the activation energy from the figure, we need to consider only two thermodynamic properties of the reaction. First, the free energy difference, short for delta G, between the products and the reactants determines whether the reaction occurs spontaneously. And the energy required to initiate the conversion of reactants into products, it determines the rate of the reaction. But the free energy change of a reaction, delta G, only provides information about the spontaneously but not the rate. So the picture shows here the free energy changes according to different values of delta G. So firstly, we can see that a reaction can take place spontaneously only if delta G is negative. Then a system is at equilibrium and no net charge can take place if delta G is 0. Then lastly, the figure shows here that a reaction cannot take place spontaneously if delta G is positive. Next, as for any reaction, we need to be able to determine delta G for an enzyme catalyze the reaction to know whether the reaction is spontaneous or requires an input of energy. To determine this important thermodynamic parameter, we need to take into account the nature of both the reactants and the products as well as their concentrations. Then we can use these following 7 equations shows up here. So we needed to pay attention to the meaning for these letters of the equations in the frame. It is important to stress that whether the delta G for a reaction is large, smaller, or the same as delta G 0 prime depends on the concentrations of the reactants and products. So most importantly, the criterion of spontaneity for a reaction is delta G, not delta G 0 prime. Then, whether enzyme could alter the equilibrium of a chemical reaction. From the figure, it showed us that enzymes accelerated the reaction rate, and the same equilibrium point is reached but much more quickly in the presence of an enzyme. So an enzyme cannot alter the laws of thermodynamics and consequently can't alter the equilibrium of a chemical reaction. Now, let us give an example for examination of equilibrium to further understand the meanings. So consider an enzyme catalyzed reaction, the conversion of substrate S into product P. Suppose that in the absence of an enzyme, the forward rate constant, short for Kf, for the conversion of S into P is 10⁻⁴ parts per second, and the reverse rate constant, short for Kr, for the conversion of P into S of -6 parts per second. So the equilibrium constant K is given by the ratio of these rate constants. You can look at the equation, shown here. So then using this equation, we can obtain the results. The equilibrium concentration of P product is 10 times that of S substrate. So whether or not enzyme is present. However, it might take a very long time to approach this equilibrium without enzyme, whereas equilibrium could be attained rapidly in the presence of a suitable enzyme. So therefore, enzymes alter only the reaction rate and not the reaction equilibrium. Here is some knowledge about today, and thank you for your attention.